Thermo's really trying to be proactive, right? So I think that's something even with Connect Enterprise, right? Making sure that it is application agnostic and making sure that we can actually prepare for the future in a way.
Welcome to Data and Biotech, a podcast from Cornell where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
John Liberty, welcome to the Data and Biotech podcast.
Thank you very much. Happy to be here.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what brings you here?
Yeah, absolutely. So my name is John Liberty. I started out my career at Charles River Laboratories. I was working in a GMP lab, primarily doing analytical services, so helping with routine testing, whether that was, something like plate-based assays and ELISA, or even just a lot of the compendial testing, like something like osmolarity or endotoxin. And then I transitioned into a startup company where I was primarily responsible for assay development surrounding PK and biomarker work. And I was also helping to outsource that, so building relationships with different CROs as we continue to push our program forward. And then following my startup experience, I moved into my current role at Thermo Fisher, where currently now I'm part of DSAS, which is the Digital Science and Automation Solutions Group. And what we do is we help work with different groups and potential customers to help implement different LIMS solutions. And now we're currently partnering with our lab automation team to help implement some of our lab automation solutions.
Can you just give us an overview of Thermo Fisher's work and then, specifically what your department does inside of Thermo Fisher?
Mm-hmm. Absolutely. So to be honest with you, I probably couldn't even give a high-level overview of Thermo in general. I mean, Thermo is anywhere and everywhere. And I think anyone who works in the science industry has heard of Thermo Fisher, whether it's in one capacity or another, whether it's getting reagents or, now that we've acquired PPD and working with some of our, BioA services or even beyond that. So Thermo is pretty much everywhere. They have their hands in every step of the process and can really help drive you from start to finish, which is really great. My group in particular, we focus on digital solutions. So helping with LIMS, or if you're not familiar, a laboratory information management system. And what that does is it can help you with sample tracking and sample inventory all the way up through data and assay management and then reporting. So in particular, I specialize in something called Watson LIMS, which is a bioanalysis specific LIMS solution.
Looking at Watson LIMS and with the context that it's bioanalysis specific, would you just give us an overview of Watson and what makes Watson sort of bioanalysis specific?
Absolutely. So it does have a lot of general capabilities that you would see in most LIMS. That can include things like sample inventory and sample management and real-time sample tracking. So it also does things like automatic freeze-thaw tracking, which is something really important, especially as you move into a more regulated environment. But then specifically when it comes to assay management and reporting and actually data management, it is very specifically tailored to BioA. And what that means is you can actually facilitate and build your bioanalysis specific protocols into the software. And then it will help support that process as you move through your workflow. And it also integrates with a lot of BioA-specific instruments, whether that's something like ELISA or HPLC or MSD. So it has those integrations in a few different capacities. You can have something what we call built-in digital interface, or we also utilize one of our other products called Integration Manager, which can help bridge the gap between any sort of instrument that doesn't have a built-in integration to Watson. So what Integration Manager is responsible for is it actually takes the data output from your instrument and it will transform it into a file that can be easily digested by Watson. So then you can actually import that data right into your study. And then it also has on the reporting side something called CDISC reporting, as well as other out-of-the-box reports, which help drive the regulatory process. So it's all specifically tailored to meet BioA specifications and guidelines as per different compliance bodies and things of that nature. And it also drives our PK, which is a pharmacokinetic module. So if you have any PK modeling, which is pretty much always necessary when you're doing drug development, especially in the pre-clin or once you move into the clinical space. So that's something that is supported in Watson as well.
Awesome. Yeah. Well, I want to dive into each segment of functionality there, the data integration, the PK modeling, all of that. But before we do, I'm just interested in when is the phase of a company's life cycle when they decide that Watson LIMS is sort of the right solution for them, or what are the challenges that they're facing at the moment when they decide, yes, this is a new tool that we need to bring in and we need to implement?
Yeah, I mean, that's a great question. And I think it's really company specific, right? So we see companies that are 20 years down the line that are currently using paper-based solutions and really have come to the crossroads of we need something more modernized and we need something a little bit more flexible. So we see companies all the time that will come to us and say, all right, how do we overhaul our current solution? These are our challenges. These are our bottlenecks. And then they want to actually implement something like Watson. Or on the other hand, we have startup companies that are, just starting to build out their program, just starting to build out their workflows, and they want to get this started from the jump. There are barriers depending on, which road you take. I think as with any type of solution in this industry, it does come with a price tag. So if you have a startup company that may be a little bit more limited on funds, that could not necessarily be realistic from the jump. I think we are working to do more startup-based modeling with our pricing so that we can actually try to get that solution implemented from an early stage, because Watson is built in a way that you can get it established and then continue to expand on it over time. It's not a one and done type of transaction. So Thermo really wants to partner with our customers to continue expanding things like the number of licenses that you have or the types of modules that we offer in our solution. So, we definitely see both sides of the coin, either doing it right in the jump or doing it, far down the line. And if you're working with a company who may be in a GMP or GLP environment and they're looking to implement something like Watson down the line, it can take a little bit of time because, as anyone who worked in a regulated environment knows, there's so many stock gaps and, so many procedures and processes and change requests and things like that to follow before you can actually completely overhaul the way that you're handling data and, logging your samples and initiating your laboratory workflows.
Yeah, I'm interested in, what that implementation process looks like if you're already coming into a GMP or a GLP environment, because, you're going from a world where, all of these processes are I'm imagining sort of triggered from like external systems and then sort of like patched together through people processes to then, integrating the data and triggering them all from like that centralized interface. Am I thinking about that right? Or how does that look?
Nope, that's exactly right. So, I think especially when you go into the current biowaste space, right, there's a lot of fragmentation that's going on. So you have all these different instruments that have their own instrument software, and they have different file outputs that may be a little bit unique. And so I think one of the greatest challenges that we see in the industry today is being able to really connect all of those different pieces, because one lab may not be doing the same assay as another type of lab. And if you want to be able to have an off-the-shelf solution, which is what we consider Watson to be, it needs to be able to handle all of these different scenarios and all these different types of output. So I think that's definitely a challenge that we see today.
Obviously, there are lots of LIMS in the LIMS space. I'm interested in even within Thermo Fisher, as I understand it, there's multiple LIMS systems that are sort of on available for organizations to purchase. So, why what differentiates Watson LIMS from some of the other LIMS that are out there in the ecosystem and in your portfolio as being sort of the LIMS that's like custom tailored for like bioanalysis CROs or bioanalysis work?
Sure. Yeah, absolutely. So I think, in general, being able to have the Thermo Fisher Scientific name behind our products really delivers a type of trust in what we actually offer. Because I think Thermo, as I mentioned, has been a staple in so many laboratories for so long that I think that really helps establish our customer base and really speak to, the types of quality of products that we're actually putting into the market. So I think that's a really great thing to have in our corner. I think also within our portfolio, we have different solutions that are tailored to different industries. So one of our other solutions called Sample Manager, it is much more versatile. It has the ability to work in different industries such as oil and gas or food and bev or manufacturing or even, large pharma. So that's something that's widely popular as well. I think where we differentiate here is that Watson is specifically built for purpose, as I've mentioned, for BioA. So anything that's not in the bioanalytical space would automatically move away from Watson. And that, again, just speaks to the types of assays that it supports, the way that it regresses data, the models that are already built into it, the reports that it generates with that data, and so on. But, we do have other products that help satisfy those requirements in other industries. And of course, with any other product, right, there's always going to be competitors, and there are a lot of other really great solutions out there. But we really try to drive home the point that we're taking what we call a one Thermo approach. So being able to gather that trust from customers that we have the LIMS solution, we have the instruments, we have the software, we have the knowledge. So really helping to distinguish ourselves from the competitors that way.
Yeah, that makes a lot of sense, both from the customer perspective, where you want to have as few suppliers, as few trusted suppliers as possible for the different pieces of equipment and process tools and software systems that you're bringing in, but also from an integration perspective, you're able to sort of quality control the whole thing from end to end when you own each aspect of the workflow that's being done. I'm wondering if we can ground it's like the workflows that are being done in Watson LIMS in sort of like either a real example or like a canned example of like from, for example, sample receipt to validated report. What are the things that people are doing inside of Watson LIMS that the system enables?
Yeah, absolutely. So, I can give you a pretty, pretty basic example of what that would kind of look like, kind of a day in the life scenario. Sure. So there are a few different ways that you could actually initiate the workflow, but I think one of the most common ways that people will do it is that you'll start by actually designing your study. And that can be done a few different ways in and of itself, right? So you have what you call a manual study design build, which is a form that's built into Watson where you can actually manually plug in, the different treatments that you're doing, your matrix, how many subjects you have, your time points, things of that nature. Or we offer what we call ISP, or import study protocol. And this would be a pre-configured Excel sheet with various different column headers or fields that you can actually drop into Watson, and then Watson will digest that and actually populate your study in the platform. And once you actually accept that study design, it'll populate all of the samples that are generated as a result of that study. And then what you'd be doing is going into the sample handling module, and you'd actually be checking in those samples. And that also could be done a few different ways, depending on if you're maybe generating these samples in-house, or if you're actually doing something like receiving a shipment from a CRO. So you can actually log different shipments that may be expected, whether it's incoming or outgoing. Or if it's something that you're generating in-house, you would actually go in and it has the option to check, is the tube cracked, or is the sample not frozen, or, what is the status of the actual sample that you may be looking at. And you can actually go to those drop-down menus and check in either one by one, or you can actually do it in a bulk scenario. And that could also be done through something like a barcode reader. So there's a few different options here. It's not really a one-size-fits-all. What we call Watson is that it is configurable but not customizable. So a lot of the functionality is out of the box available to you, but how you tailor it to meet your needs and meet your workflows is pretty specific. So once your samples are all checked in, that's when you go and actually create a master assay. So this would be in the sense of like if you already had something validated and you're ready to do a routine analysis. You would actually go in to create a master assay, which is essentially your protocol for your method. And you would actually specify, okay, is this an ELISA? Is this an LCMS? Is it an HPLC? Or so on. And you would specify that, and then you would actually link any instruments that are integrated with the platform. And then that is when you would actually go in and set your parameters. So you would set, your different standards or your QCs and what those levels and numbers of replicates are. You would specify your regression models and, where you want your cut points to be, where you want it to throw different flags if you have percent CVs that fall out of spec as per maybe your internal SOPs. And then once you actually save that method, you can create a local run assay. And that is where you essentially take the template that you just created and just add your samples to it. So you would designate what samples belong in that method, and then you would save that as what we call an analytical run. Once the analytical run was already set up, then that is when the scientist would go into the lab and actually perform that experiment. Once you have the output from the instrument in the lab, then that's when you'd be using something like Integration Manager or the built-in digital gateway to actually move that data file from your instrument into Watson. And then once that data is in Watson and actually linked to your study, that's when you're actually going to be running your regressions. So you have the option to actually go through and view any sort of graphs or tables that may be available out of the box. So as a user, you have the option to go in, do a quick sweep of your data if you want, and then maybe pass it off to QA. So you can have Watson throw different flags and create very clear lines in the sand of where something may have fallen out of spec. And then once that review is complete, you can either accept or reject your run. If you accept your run, then that's where you could actually go in and start populating those reports. And those reports can all be exported either to like Excel or Word or something else, or downloaded locally as a PDF or however you need it to function. Then that's pretty much it. So those samples will be updated in inventory in real time if you have aliquots that are dedicated to those assays, or so on. In the case of doing something like validation, you would create a master assay that is specified as a validation run. And what that would do is that would allow you to actually pull those validation parameters like linearity, specificity, repeatability on those reports at the end. So if you had a validated master assay already, then you could use that for routine testing. If that's something that you're looking to establish early on, you would make sure that it's specified as a validation run. And then that is where all those parameters would come into place once you start pulling the data together.
Yeah, so I can see how the system is sort of templatized the entire process from end to end, and I would imagine that it reminds me of like ERP implementations that we've done in the past, where you're sort of you're taking a system that's designed to like consume a company's entire business process from end to end and make it conform to this to this sort of like outline, this data model, this way of collecting information and making sure that it integrates together. And one of the challenges that we always see, like with implementing a system like that, is that any company that's adopting the system has to sort of meet the system halfway, or maybe even more than halfway, has to make their business process like conform to the system in order to get the benefits of the automation, the integration, the process, like the efficiencies, the repeatability. So I'm just interested from your perspective in how Thermo Fisher navigates that, and like what the conversations with clients are like when trying to like make the business processes conform.
Yeah, absolutely. I think, as you mentioned, right, there's a lot of input that's really needed from the customer to figure out, one, what solution is actually right for that customer, right? Because as you mentioned, our portfolio does extend across multiple industries and a few different workflows there. So really identifying first and foremost, what solution are you targeting? And then we have what we call discovery conversations, where we're actually doing requirements gathering. And what our rule of thumb is, in my role as what we call technical sales consultants, is our biggest thing is nailing the discovery conversation. So what that looks like is a lot of active listening. We're maybe asking some questions to kind of probe some information, but for the most part, we're really just letting the customer talk. It's really just about listening to, what is working for them, what is not working for them. What are their goals for today, tomorrow, 5 years from now? And how do we really get them there? So I think, getting a lot of that information out of the gate helps us understand what we have to offer them. And I think it also really helps us prioritize what we need to deliver to them in order for them to invest in our solution. And as I mentioned earlier, right, we're not really looking to just sell something and then essentially drop off. We're looking to really establish these partnerships that we can continue to grow, because all of our solutions in our portfolio are built in a way that can be expanded. And so, we've even worked with earlier startup companies that maybe get absolute baseline Watson. Maybe you have 2-3 licenses at most. You don't have any added modules. You just kind of want to be able to log samples, create a concentration-based data regression, and maybe spit out some R&D level reporting. But then they'll come back to us after several years and say, okay, I want to add IRM, which that is our immune response module, which is an optional add-on that specializes in ADA or NAB assays. So once they really get to that step in the process, right, you can easily tack that on to your environment because the foundation of Watson is already there for you. So I think really the biggest thing is making sure that you hit discovery early and often, and making sure that you're not moving forward with the solution until you fully understand the requirements that are being laid out for you.
Yeah, that makes a lot of sense. Another thing that I'm imagining comes up that sort of relates to what you were saying about like integration earlier is that, if not all of your systems are Thermo Fisher systems, or if there's, assays that you're using that are sort of off the beaten path, or using machinery that hasn't been integrated with Watson yet. I'm just interested in, for those companies that are coming with systems or assays that don't directly integrate with Watson, how does that conversation go and how does it work?
Yep, absolutely. And that's something that's so relevant to today because science, as we know, is continuing to evolve, and it's actually accelerating in their involvement or evolvement. So I think, we definitely have those conversations where maybe some customers will come to us and say, all right, this is what I'm looking to do. This is this new system and instrument that I have. What can you do for us? And something that we've never seen before, right? That's going to happen. It's inevitable. But I think what you do is you really need to understand what they're looking to actually bring into the Watson platform. And more often than not, we're able to work with them through our Integration Manager tool. So Integration Manager, as I mentioned, is actually instrument agnostic. So it's really meant to be able to bring in data from almost anything because it functions on a customized basis. And it functions in what we call transforms. And transforms are primarily built by our services team, who are excellent. And what it does is that is the transform that is responsible for converting a data output into, primarily it's an XML file that is then ingested by Watson. So we could do it with something that we've never seen before, but there are some limitations. So, I wouldn't be truthful if I didn't say that there are some scenarios that we've come across that maybe, okay, we can't integrate with that. Sometimes it just can't be done. However, more often than not, we're able to pull pull what's being needed into the platform. And we have found a lot of workarounds around that. Because, I mean, Watson as a product, it's been around for more than 20 years. So, it definitely does need to continue to evolve with the times. And I think it's really great that we've been able to leverage this integration manager tool, because no product from 20 years ago would ever be able to have the forward thinking and the types of things that we're doing today. So I think it's nice that we have that product to fall back on to, help get us there and bridge that gap.
Do you have a sense of, you mentioned that there are situations where you can't integrate. So the integration manager tool makes a lot of sense. Like they're for systems that are able to produce data in specific output formats or in certain structures. The transforms just converted into XML and then ingested in the system, and then you can work with it inside of the Watson LIMS. I'm just interested in, what are the characteristics of the systems that make it difficult for Watson and the integration manager to work with?
Mm-hmm. Yeah, so I think a lot of it is really just about, how the data is organized. So if you have a different file output and it has, certain headers or categories, it really needs to be able to map to something that can be recognized by Watson. So that's the biggest challenge, is really just mapping those categories of data into something that is comparable in Watson. So I think our services team spends a lot of time really figuring out and picking apart those different output files to make sure that, if someone wants to get certain values in, that can actually resonate with something that Watson already has built in functionality for it.
Right. So it's like it's schema matching between what gets outputted from the system to what's in the Watson system, and making sure that those mappings are both logical to Watson as a system and logical to the customer in terms of being able to interpret the data that's being ingested and is able to go along for the ride of the entire thing. Right?
Exactly. Yep. That's exactly right.
Yeah, that makes a lot of sense. So you've got all of this sample metadata, you've got all of your experimental metadata, you've got information about the assays that you're using and the systems that you're using. At this point, you've got all of the metadata and then the data from the particular experimental runs, analytical runs that you mentioned coming in. You've mentioned a couple times the different types of modeling that bioanalytical organizations are doing inside of Watson. Would you mind just talking a little bit about the types of modeling you see customers do and what the use case for those models are? And I understand that people who are sort of indoctrinated into the biotech ecosystem might already know what they are, but our podcast has people at sort of varying degrees of exposure to biotech.
Absolutely. I think if you're doing something a little bit more mainstream, like maybe even ELISA, you might be using something like a 4PL or 4PL quadratic curve to do regressions for concentration-based analysis. And then if you're doing something like qPCR, that's something that might more tailor towards a semi-log regression model. Those are just two examples of things that are pretty common in the.
Can you just talk about what are the questions that people are trying to answer with those, and then how Watson sort of answers those questions, or
Yeah, I can more so speak to the concentration based one, but what you'd be doing there is be taking something maybe a raw absorbance value from your plate reader and actually putting it into Watson, and what that would do is it will create a regression based off the model like the 4PL, where you're actually solving for unknown concentrations of your samples. So you have your standard curve with your QCs and checks in place, and then you'll actually be getting those concentration values for your unknown samples, and then you can actually use that to create those flags and make sure that if you're doing something like a validated run, that all of those checks and balances are being met by something like QCs and percent recoveries and CVs and things of that nature.
Yeah, so the plate has different concentrations in it, and you're using those different concentrations to sort of estimate how absorption occurs at different degrees of concentration, that sort of thing.
Yep, exactly. And so even in the case of an ELISA, you could have an 8-point curve where one could potentially be a concentration of 0. And you're actually graphing all of those standard points at known concentrations, and then it will interpolate from that curve an unknown absorbance value and spit out a concentration value.
So downstream of that experiment, how is that going into decision? What are the types of decisions that either the bioanalysis organization or the downstream like customer of the bioanalysis organization is trying to make using like the outputs of that.
Sure. So I could give you one very concrete example, which would be something like if you're doing like a preclinical study and you're trying to figure out your dosing for a potential new drug that you're trialing, right? You're not sure exactly what your ROA or your route of administration would be. You're not sure how high to dose the drug in order to get the efficacy that you're looking for. I think first and foremost, you need to understand what concentration is actually making it to the target system. So that's where a lot of that comes into play. In particular, in my experience at the startup company, right, we were doing a drug development for neurodegeneration. So I was doing a lot of testing on different brain tissue and trying to understand what concentration of our drug actually made it to that designated part of the brain. So otherwise, you may need to adjust your dosing, or you may need to adjust your route of administration, because you really need to make sure that your target drug is reaching the molecules that it needs to and connecting through the intended pathways to get the desired clinical outcome.
Yeah, that makes a lot of sense. And another thing I heard you talk about a little bit earlier was this idea of like testing for immunogenicity or for like antibodies. And my understanding is that Watson sort of helps with modeling that as well. Am I remembering correctly?
Yep, that's absolutely correct. So that is like the added IRM or immune response module that we have for our environment. So as you mentioned, that is specific to ADA or anti-drug antibody and then NAB or neutralizing antibody assays. And so what that does is in a similar way to the baseline Watson, it helps really guide users through the process. So specifically with ADA, right, it functions on a 3-tier methodology. It goes through a screening tier, then a confirmation tier, and then a titer tier. So what that really means is first you would take a sample and you would actually screen and say, okay, are there anti-drug antibodies present in this sample? Yes or no? If it's yes, then you'd actually move to your confirmation assay, where you want to make sure that there's no false positives from the screen. And that could be from a variety of different reasons. But you want to make sure that you're actually confirming that. And what they do is they actually immunodeplete those samples in a way that confirms if those antibodies are actually present or not. And then if you move on to the third tier, that would be the titer tier, where you're actually quantifying how many antibodies have formed as a result of your drug.
Right.
And that's something really important because you want to make sure that if there are any potential negative effects as a result of those antibodies, or if the antibodies are actually inhibiting any of the effects that you're hoping to get from the drug.
Right. And my understanding is that there's sort of, if I'm remembering correctly, there's like a cut point analysis of like, where like sort of the limits to what you can, what you believe that your patients can tolerate in terms of the amount of anti-drug antibodies or like immunogenicity at like different degrees of like dosing and concentration. Am I thinking about that right? Or like,
Yep. Yeah, that's absolutely right. And also like cut points will be established to determine if something really does fall in a positive or negative round.
Right. Well, you talked about like the integration module, but I know that there's this idea of like Connect Enterprise, this sort of unified platform vision for where Thermo Fisher is going. Can you maybe outline that for me?
Yeah, absolutely. And this is there's a lot to this. So I'm going to try to break it down into steps that I think are a little bit more palatable here. But please, just to set the stage, Integration Manager is something that is a Thermo product, as I've mentioned, that helps transform data. And it is specific to some of our current solutions like Sample Manager. You can use it on occasion, or Watson. A lot of our customers for Watson use it. Connect Enterprise functions on something completely different. So Connect Enterprise is a brand new up and coming platform that Thermo is working on that is essentially meant to create a single UI across all of your current applications and really just enhance your digital ecosystem. And what's really powerful about that is that it is application agnostic. So it can function with Thermo Fisher applications or third-party applications and softwares alike. So the goal here is to not really replace anything that you're currently using. It's really just meant to enhance your digital ecosystem. So if you have something that you're using, maybe like a third-party scheduler tool to help your lab staff schedule when instruments are going to be in use, and then you have your LIMS, and then you have your ELN or your electronic lab notebook, and then you have your reporting tools. Those may be all different solutions. So we see all the time that customers are using a wide variety of different applications. And we definitely know too, even though Thermo Fisher is everywhere, there's no one who's using exclusively Thermo Fisher. So we thought, okay, how can we actually capitalize on that? And that's where we came up with Connect Enterprise, where we're actually able to pull in the UI and the functionality of all these different solutions, regardless of if they're Thermo or not. So what you do is it functions on primarily APIs, but then we also have what we call Connect Transfer. And there's a lot of connect here, so it can get a little confusing. But the way I describe Connect Transfer is it functions similarly to Integration Manager, but it is exclusively for Connect. So through API and through Connect Transfer, you can actually link all your applications to a single login on Connect Enterprise platform. And you can create dashboards and reports that are, potentially cross application information. So if you wanted to initiate a workflow that, you pull in sample information and metadata from your LIMS, but you want to simultaneously create a new lab notebook entry, and you want to leverage your scheduler tool, you can essentially log into Connect Enterprise once it's set up and access all three of those platforms through the UI of Connect Enterprise, and then it will all sync in real time.
Interesting. Okay, let me try to see if I can understand.
It's a lot.
What's going on, because Watson. So there's multiple levels of data integration happening here. There's the level of data integration where, when you're doing a Watson LIMS implementation, you're sort of orchestrating the entire bioanalytical workflow from end to end within Watson LIMS. But then there's still going to be software systems that are outside of the scope of sort of that end-to-end workflow. So you mentioned reporting systems or orchestration systems or something like that. Anything where information needs to move from one system to another, or there needs to be a higher level of either triggering of work or movement of data. And, maybe you want like a single sign-on or a single place where where you can log into all of those systems and I don't know. That's the part that doesn't necessarily make sense to me, because my understanding is that Watson LIMS is doing this orchestration, but then there's also this opportunity to do it in Connect Enterprise. So maybe if you can help to distinguish when a system would be integrated with Watson or through Integration Manager, and when Connect Enterprise would sort of be the umbrella it falls under.
Yeah, absolutely. So Connect Enterprise is really, someone used this analogy the other day, and I actually really liked it. So I think I'm gonna recycle it. But Connect Enterprise can serve really as the mortar to the brick wall of your digital ecosystem. So it really can be in between all your different systems. So in a perfect world, if you have a solution like Sample Manager LIMS, you can actually link your Sample Manager LIMS to your Connect Enterprise platform. And then if you're using something like, Chromeleon or some other like lab automation system, like in Thermo, we have another lab automation tool called Momentum. So if you're using that, what you can do is create those workflows in Connect Enterprise and then pull metadata in real time from Sample Manager, and then initiate your workflow in something like Momentum or whatever else you're using. Maybe you're running an LCMS assay on Chromeleon or, what have you. And it'll actually be updating and pulling reports in real time, and it will actually send that data back out to their home applications. So if you were to log into Sample Manager on your own after you initiated something, it'll actually be updated and populated in real time, because Connect Enterprise will be pushing that data back into Sample Manager, but it'll also be available in Connect Enterprise. So you don't actually have to go back into Sample Manager if you didn't want to. And you can actually pull that information in. So Watson and Sample Manager, as they stand today, they're just LIMS systems. Whereas Connect is supposed to be everything.
Yeah.
It can tailor your LIMS system and pull it in and connect to your electronic lab notebook or your lab scheduler or any other types of reporting tools that you may have.
Let me say back to you what I'm understanding. So they're just LIMS systems, they're just laboratory information management systems, which is, a place where you house data, where the outputs of all of these different workflows and the metadata from all of these different from all of these different machines and process steps are coming together so that you can answer the questions, generate the reports that you need for regulatory compliance and whatnot. But if you want to take a step toward full scale lab automation, where you're able to connect the dots between all sorts of different systems, including your LIMS systems, your ELN system, your lab automation system, if you want to connect those dots, then you need something like Connect Enterprise.
Exactly. I think the larger picture here is that Connect Enterprise, the scope of that product goes so far beyond just the LIMS. And that's the biggest thing here, is that, the LIMS has its own scope of what it's meant to do and how it helps, drive your workflow. But anything beyond what a LIMS can do is meant to be caught by Connect Enterprise and really just link everything together in one place. Because I think something that, I've heard talked a lot about is application fatigue, right? It's so real. It is something all the time. Well, even when we're talking about Connect Enterprise with customers, right? The last thing people want to hear is, hey, you should go get this new product. You should go get this additional platform and get all this training on something completely new. I completely understand. It is so true. I think, as I mentioned earlier on in the conversation, right, with the fragmentation, people are responsible for learning and exploring and training on a wide variety of different applications and softwares and the ins and outs of everything. So I think Connect really is meant to be a solution to that, because if you can bridge everything together into that one Connect Enterprise ecosystem, you won't necessarily have to go manually into each individual application. That is like the larger picture here.
Yeah, that makes sense. And if I may editorialize for a moment, like one of the things that I've heard is also that it's not clear where the boundaries of a LIMS system or an ELN like end and begin, or lab automation system, each of these systems sort of has its lane, but the lanes overlap with each other. And so, we've got these puzzle pieces that don't always it's not always clear which piece you should use for which segment of what the organization is doing. And so then when it comes to like thinking about like tying them all together across an entire across an industry where different companies are using systems for different overlapping purposes, it's it's understandably hard to imagine like how it all fits together. But that's sort of like how it looks from the outside. Would be interested on your take on how it is on the inside.
Yeah, absolutely. I mean, I think that's very true. And even if you were to pull some information from something like Sample Manager or different competitors or things like that, you could potentially get different answers, because I think people are continuously adding functionality and features to their product to try to expand into those other lanes that they initially intended to not be a part of. So I think the scope of different products can definitely vary greatly. It really could be subject to the use cases of where one product starts and where one product ends.
In an ideal world, like you're helping to architect a startup lab from scratch. Let's say we've got a startup biotech from scratch. We've got a startup biotech that's trying to establish itself with the right data and process and regulatory infrastructure. And it's a small biotech that's expecting to raise funding, expecting to grow quickly. I would love to just hear from you, like, how would you guide an organization like that in sort of the life cycle of their, like, of when they need which piece of like a software, and we could use like the software the systems that Thermo offers. Like, when should they be thinking about tacking on each element of it, and then where should they be looking as they scale into like the actual enterprise? They've gone public and they're now ready to put on their big boy big girl pants and go be a full-fledged biotech.
Sure. Yeah. So in my opinion, I think one of the best first steps to take is really the lab notebook, whether that's an electronic lab notebook or otherwise. But I think really that's where everything starts, right? If you're trying to do assay development from scratch and you're really trying to get your experiments up the ground, that's really where you're going to begin. So having an ELN that can easily organize your data and initiate different reviews and make sure that it's stored in a way that is reachable and you can easily recall it and things of that nature. Once you start scaling up and you get more samples in your lab and you're kind of have potentially different partnerships where you're working off site or you're doing shipments to and from a CRO, that is where I think the LIMS comes into play, because it can be really easy to lose track of samples when you don't actually have a system in play. You see, any scientist will tell you, right? You go into a -80 freezer and you may have started with the idea of having all these designated racks for storage and labels and all that. But I mean, all it takes is something like a label to fall off a rack because it's -80 degrees Celsius. And then you lose track of the sample, right? So it can be really easy to lose that sense of organization, especially once all the frost starts coming into the freezer and, you need to de-ice it and all that. So I think that's really where a LIMS comes into place, is when you start scaling up the number of samples that you have, and you need to start really organizing your data in a way that can be prepped for a regulatory body. And also, once you start expanding headcount, you really need to make sure that data is available to all the people who need access to it. And that's where something like a LIMS could actually help facilitate that transition from person to person, and also make sure that data is readable and easily reviewable. Once you have all of those other applications in play, that is where Connect Enterprise would come into place. Because as it stands today, Connect Enterprise is not an out-of-the-box solution. I talk about Watson as an out-of-the-box solution, meaning that you install it, you get it up and running in your ecosystem, and that functionality that is built into the product is available to you from day one. Whereas Connect Enterprise is really meant to be a fully customized implementation. So we really need to see what you're working with for applications. How are you actually logging in samples? How are you keeping track of your laboratory entries? How are you actually handling data storage and data maintenance? And then what we do is we actually build a solution to tailor that. And that's kind of where I mentioned where it's kind of like the mortar and the brick, because you have the bricks already in play, and then Connect Enterprise helps fill all the gaps between that and hold it all together. So I think Connect Enterprise would really be the last piece of the puzzle in those ecosystems. And once you really get up and running and you have all your applications working on a regular basis, that is where you can actually drive them into the future with Connect.
Yeah, that makes a lot of sense. It's like one of the reasons why Connect Enterprise seems so amorphous is because, the mortar like takes a different shape depending on which bricks you're using, and it sounds like it's highly customizable and then it's highly specific to the organization that's being implemented.
Yep, that's exactly right. So I think even we have people who are going on site to potential Connect Enterprise customers and doing workshops and POCs or proof of concepts, and that is really where it comes into play, is like having something that's already up and running, and how can we make it better? How can we make it more easily to communicate across different applications, and how can we continue expanding this so that as science continues to accelerate, we can keep up with it and get ahead of it.
I'm interested in how you demonstrate the ROI of Connect Enterprise to the companies that are adopting it. I mean, obviously, like regulatory compliance is like a clear win. It's something that has to be done. But then there's also sort of like the collaboration gains that you mentioned, and then there's also like sort of the reliability, the repeatability, the reproducibility of the things that are happening. So I'm just interested in what are the ROI stories that you think resonate most with the customers that you're talking to?
Yeah, sure. So I think those are two. First of all, I think those are two totally different conversations, right? Because an ROI on something like Watson would be completely different than the ROI on something that or a company that would be looking to implement Connect Enterprise. So I think with Watson, as you mentioned, regulatory compliance is a really big thing. I think in general, we see a lot of customers who may be looking to move into the GLP space, or actually maybe looking to file their IND papers and things like that. And that is where you really need to start getting into a solution that is tailored to that and is really built to promote that compliance throughout, whether that's, CFR, electronic signatures, or, FDA guidance and things like that. So that would really be the biggest payoff if you're moving from a non-regulated to a regulated space with Watson. I think something with Connect, we're actually working internally to figure out how to present that story in the best way. I think something that's really interesting is we're actually working to build out what we call the new Customer Experience Center. And that is a location that we have where we have a variety of workflows initiated by our lab automation group and our Momentum software. And we have it already integrated with our Connect Enterprise platform. And we're actually hoping to continue bringing prospective customers on site so that they can actually see it in action. And what that does is it really showcases the lab automation capabilities while also showing how Connect drives that process. So even I've been part of one conversation with prospective customers with this, what we call the CXC, Customer Experience Center. And what it does is you actually initiate your workflow that's pre-configured in Connect. You click go, essentially. And what it can do is it can pull that information from Sample Manager for your sample metadata. Then you'll actually move into the wet lab, and you'll actually see our autonomous robots grabbing plates and aliquoting different samples, and then running things like an IC50 assay or an LCMS on our Vanquish. And then it will actually use Connect Transfer, as I mentioned, to pull that data back into Connect Enterprise, where you can actually populate the reports. So what you see is fully autonomous lab with minimal human intervention. And what we really try to drive with that messaging is that if you have a workflow that's already solidified, you can just implement the lab automation piece to essentially have that on loop. And you can skyrocket your capabilities by having something run on loop overnight with minimal human intervention. And the goal here is not to replace human intervention. It's really just to change the focus on where the human intervention comes into play, right? Because if you have robots doing something like aliquoting or placing a plate on an IC50 plate reader, you can actually have your scientists focusing on other method development or continuing to drive your portfolio forward. So it's really just shifting your resources into, where is the best use of your time, and how can we eliminate the need for manpower on, low value or highly repetitive tasks. So that's the ROI that we're trying to paint with that CXC.
Yeah, and sort of the facility that you mentioned with fully autonomous labs in the future. I would love to. I would love to see that at some point. That's pretty. That sounds pretty amazing. It sounds like a good way of showing, not telling people what you're driving toward, which seems like the only approach that's possible when you're talking about something as amorphous as connecting.
Absolutely. And I think especially in today's day and age too, right? We see people all the time getting on Teams calls or Zoom calls and seeing things. And it's kind of like, oh, okay, well, that's cool. But at the same time, when you're actually there, when you're physically seeing these, even what we call an AMR, an autonomous mobile robot, actually moving from lab to lab, room to room, picking up plates, aliquoting samples, actually, moving those data files from place to place, it's really quite something. And I think it really does paint a really powerful message of what the capabilities are for scaling up if you have something moving like that and seeing it in real time. I think it really just drives the message home by being able to be there with it and really see how it works and get a glimpse under the hood of how you could potentially adopt this in your own system.
Yeah, and from a data science perspective, we're always interested in having more data, and you hear about sort of the foundation models for biology and how, they're still relatively data constrained, and this idea that, lab automation, that autonomous labs could, also increase the throughput of the data that you're able to gather, of the number of experiments that you're able to run, and thereby, in some combination with machine learning and AI, be able to derive much better insights, or be able to, optimize a compound for, bringing it to market much more quickly. That seems to be where a lot of the conversation is going. So that makes a lot of sense. To summarize that, it kind of feels like we're moving from a world that's more instrument centric to one that's more data centric. Would you agree or disagree with that idea? And how would you characterize it?
I think that's definitely accurate. I think data is the biggest, the front and center concern that a lot of people have of, how are we maintaining data security and how are we actually tackling data storage and making sure that, things like audit trails are being captured and, everything that's really needed in a regulated environment. So I think being able to create solutions like Connect that is centered around data transfer and data storage, and I think making sure that we can actually keep up with the current evolvement of science. And I think there's always new instruments and always new softwares and data types that are being generated and starting to flood the industry and the markets. So making sure that we're really just keeping up with the direction the data is going in. Because once the data is out there, you need solutions that can actually tackle it and store it and manage it in a way that is going to be beneficial to the users.
The ecosystem is so broad and evolving so quickly. And so, what I hear you talking about with Integration Manager and with Connect Enterprise is this idea that that Thermo Fisher is trying to create the abstractions, the primitives that allow you all to interface with the broader digital science ecosystem and bring all of this data together. I'm just interested in, where do you think Thermo Fisher's role is in that environment? And then how do you continue to stay abreast of where customers are going in terms of the tools that they're using and the data that they're trying to bring into that system?
Yeah, I think, Thermo's really trying to be proactive, right? So I think that's something even with Connect Enterprise, right? Making sure that it is application agnostic and making sure that we can actually prepare for the future in a way, because we're not going to be able to predict what is coming down the line in 1, 2, 3, 5 years from now. So making sure that we can create solutions that are, here for the long term for our customers. So I think Thermo's been doing or having a really large push in creating something that can serve you today, but then also serve you in 10 years from now.
There is seemingly infinite space for new drugs or new therapies to come to market and help improve people's lives. But making the economics of the entire ecosystem make more sense from a funding perspective, I think, has the potential to create even more opportunities for science and raise all boats. I don't know if you have any thoughts on that.
Yeah, absolutely. I mean, I think you mentioned, right, scientists, if you asked any scientists off the street, I feel like more often than not, they're going to tell you that, doing something like a repetitive task of pipetting or routine analysis for, sample prep isn't necessarily something that they want to be doing all the time. I mean, it can definitely be, new and exciting, especially as you're doing something like assay development. You're doing trial and error. That's the basis of science and the scientific method. That's what's really exciting. Once you get something solidified and validated, if we can actually have things like lab automation tools that can do the sample prep or the early on, low-hanging fruit or something like that, then you can actually have the scientists working on the more innovative components of it and really be on the back end of the things that really can bring value to the company and to the group.
That makes a lot of sense. So as we head toward the end, where can people go to learn more about you?
Yeah, absolutely. So feel free to add me on LinkedIn or shoot me a message on there. Again, my name is John Livery. And also we have lots of information and materials available on our Thermo Fisher Scientific website. So in particular, if you're interested in a LIMS like Sample Manager or Watson, or interested in starting conversations about our new and upcoming Connect platform, feel free to shoot right to the Thermo Fisher Scientific website, and you should be able to find what you're looking for. And if anyone wants to shoot me an email, feel free to connect. My email is john.liberty@thermofisher.com.
Awesome. Well, John, it's been a pleasure having you on the podcast. Really appreciate it, and look forward to connecting down the line.
Thank you very much. It's been great to be here.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.